Welcome back to AI and Stuff, where the future is, apparently, a lab assistant that reads every paper, never asks for coffee, and still needs humans to check the results. Today's story is not AI cured cancer. It is better than that, because it is real. The real story is AI is starting to help scientists find better research leads faster. Nature published a paper on May 19, 2026 called Accelerating Scientific Discovery with Co-Scientist. Co-Scientist is a multi-agent AI system from Google designed to work with scientists as a research partner. That is the key word, partner. Not replacement, not magic doctor, not a chatbot wearing a stethoscope it bought online. This system is built to help humans do the hardest early part of science, deciding which idea is worth testing next. Instead of only summarizing papers, Co-Scientist generates hypotheses, critiques them, ranks them, debates them, and improves them. Imagine a tiny scientific team inside a computer. One AI agent says, "Here is a possible drug idea." Another agent says, "Interesting, but prove it." A ranking agent compares the ideas, and the strongest ones move forward. It's like peer review at super speed, except nobody has to pretend the conference sandwich was good. The first major example was acute myeloid leukemia, or AML. AML is an aggressive blood cancer where abnormal white blood cells multiply quickly and crowd out healthy blood cells.
The researchers asked Co-Scientist for drug repurposing ideas. Drug repurposing means taking an existing drug and asking, could this help with a different disease? That matters because existing drugs may already have known manufacturing, dosing, or safety information. So AI is not just inventing random molecules in the dark. It is helping scientists search the giant library of what already exists. In AML cell line tests, 3 expert-selected candidates stood out: binimetinib, pacranitinib, and cerevostatin. The paper reports that binimetinib showed an IC50 as low as 7 nanomolar in AML cell lines. Simple translation, in a lab dish, a very small amount was enough to reduce cancer cell viability by half. That is an early signal, but it is a useful signal. And this is where AI becomes powerful. It can scan more connections than one human team can comfortably hold in their heads. The next result was even more interesting. Co-scientists suggested KIRA6, an IRE1 alpha inhibitor, as a novel AML repurposing candidate. In cell line tests, KIRA6 inhibited KG1, MOLM13, and HL60 AML cells. The IC50 values were 13 nanomolar for KG1, 517 nanomolar for MOLM13, and 817 nanomolar for HL60. In normal human language, AI helped researchers toward a non-obvious lead, and the first lab check showed measurable activity. That is exactly the kind of job AI should be doing. Find the lead, show the evidence, let scientists test it. Now, because we are adults and not comment section fireworks, we still need the caveat. Cell experiments do not automatically become patient treatments. The paper itself says in vitro results do not guarantee animal results, clinical success, safety, dosing, or patient benefit. But that does not make the results small. Early science is a funnel.
Most ideas fail. AI's promise is that it can help humans put better ideas into the top of that funnel. The second example was liver fibrosis. Liver fibrosis is scarring in the liver, and serious cases can lead to liver failure or liver cancer. Co-scientists searched for epigenetic targets. Epigenetics is like sticky notes on DNA. The recipe book stays the same, but the sticky notes tell the cell which recipes to read more or less. Researchers tested drugs connected to AI-suggested targets in human hepatic organoids. Organoids are tiny lab-grown tissue models, basically practice fields for biology. The paper says drugs targeting 2 of the 3 AI-suggested epigenetic modifiers showed significant antifibrotic activity without causing cellular toxicity in that model. Again, not a prescription, but it is a sharper map. And in science, a sharper map can save years. The third example moves from cancer to antibiotic resistance. The focus was CFPICIs, mobile genetic elements that can help move virulence and antibiotic resistance genes between bacteria. Researchers already had an unpublished experimental finding about how these elements spread. They asked co-scientists to reason from public literature and propose the mechanism. The AI's top hypothesis matched the key idea. CFPICIs may interact with diverse phage tails to expand their host range. The paper says the AI generated this matching hypothesis in 2 days. That does not mean AI created knowledge from nowhere. It means AI connected public clues at a speed that could make scientific exploration much faster. And that is the pro-AI point here. The best AI systems are not replacing careful science. They are making careful science more powerful. They can read widely, rank ideas, suggest experiments, and help humans spend their limited lab time on the most promising leads. Where this is headed is a new research workflow. Scientists ask better questions. AI searches the maze. The lab tests the leads. The results feed back into the next round. That loop could speed up drug repurposing, rare disease research, antibiotic resistance work, and fields where the literature is already too big for any one person to hold.
So no, AI did not cure cancer. But AI is becoming a serious research accelerator. It helped find cancer drug clues, liver fibrosis targets, and an antimicrobial-resistance mechanism. Some clues survived real lab checks. That is not hype. That is what useful AI looks like. Not replacing scientists, giving scientists better searchlights.